How has the integration of mass spec impacted your workflow in your cancer studies? And what are some of the most critical analytical challenges you face when profiling these tumors?
Yeah, those are great questions. So I've utilized mass spec from very basic science mechanism stuff to using it to actually study human tumors grown into mice. So we have used it on the basic science side of things to answer questions like what specific transcription factor is binding this sequence of DNA in the promoter region of Notch3 to drive this tumor suppressor in neuroendocrine cancer. I have answered that question with mass spec, which is very niche, but also very important. But at the same time, we use mass spec on, we put human neuroendocrine cancer cells into a mouse that didn't have an immune system, of course, grew those tumors, and actually had a couple groups of tumors. Some that were just wild type, and then some I had used CRISPR to engineer some of the tumor cells and then put these different groups into mice and then saw how they grew differently or how they behaved differently. And part of the analysis was using mass spec to identify proteins that were differentially expressed between these different groups of neuroendocrine tumor cells that I had engineered with CRISPR. And so we were able to find differential protein expression. Actually, it'll be published soon that I can, hopefully soon, share with everyone our exciting findings on what was different between those two groups. And so mass spec has been a excellent tool for me in my studies.